In this video, we're going to take a closer look at the small intestines. The functions of the small intestines include completing the process of food digestion, absorbing nutrients, and there are some secretions that occur. If we trace the path of chyme through the small intestines, it starts at the duodenum, which receives the chyme from the stomach. From the duodenum, chyme travels to the jejunum, and then lastly to the ileum. The undigested food product from the ileum will then go into the large intestine, specifically the cecum. The wall of the small intestine is similar to the wall of the stomach, the wall of the esophagus, and any other gastrointestinal wall that we've looked at. It has the same four layers: the mucosa, submucosa, muscularis externa, and serosa. So the mucosa is the layer that's in contact with food. It's the layer facing the lumen. The mucosa has three parts to it. The epithelium, which is the actual portion of the mucosa that faces the lumen and has contact with food. And then next to that is a layer of connective tissue called the lamina propria. And then next to that is the muscularis mucosae. We're going to look at some specializations of the small intestinal wall, specifically specializations of the mucosa. The small intestine mucosa is highly specialized, okay? And basically most of the specialization comes in the form of increasing the surface area for absorption of nutrients. We absorb nutrients by making contact with those nutrients. So if we can increase the surface area for contacting nutrients, then we can increase the surface area for absorption. And we do this basically by having folds of the mucosa with folds of the mucosa with other folds, right? Folds within folds within folds. All of these different folds, like I said, increase the surface area for absorption. But let's take a look at some of the specific specializations. Firstly, the epithelium of the mucosa is made of simple columnar epithelial cells. Sometimes these cells are called brush border cells, and that's often how I will refer to them, but you can call them absorptive cells or enterocytes or small intestinal cells. They're pretty much all the same thing. We call them brush border cells because they kind of look like they have a little brush on the side facing the lumen, and that brush is made of microvilli. The job of the brush border cells is to release brush border enzymes, which help to complete the process of digestion. And then the microvilli facing the lumen are going to function in absorbing the nutrients. In addition to these brush border cells or simple columnar epithelial cells, there are also mucus cells, sometimes called goblet cells, which are located embedded in the epithelium, and of course they produce mucus. There are also going to be enteroendocrine cells, or just endocrine cells if you will, which will be producing hormones. So these are also located in the epithelium, and they are going to produce many hormones such as CCK, secretin, and there's many more. If we look on the other side of the simple columnar epithelium, we'll find that lamina propria. So this is a layer of connective tissue, but it does contain capillaries and lacteals. Both of these are going to be important for transporting absorbed nutrients. So both the blood and the lymph will carry transport absorbed substances away from the small intestine. Take a look at the images here. We can see how we have folds in the small intestine itself that we can actually see with our naked eye. So gross anatomical folds, and those are called circular folds or plicae. So that's one way to increase the surface area. But when we look at these circular folds or plicae, we see that they also have folds within them called villi. When we look at the epithelium coating the outside of the villi, we can see more folds in the form of microvilli. So all of these help to serve to increase that surface area for absorption. The villi and microvilli are part of the mucosa, but the plicae are actually part of the submucosa. So that leads me to my next topic, which is specializations of the submucosa. And so I basically already said this on the previous slide, but the specialization of the submucosa is to form those larger folds called plicae. Plica for singular, plicae for plural. And in this picture here, if you look at number 19, that's our duodenum there, and it kind of looks like an accordion, right? So those folds are the plesae. And they're actually kind of interesting because they're in a spiral, and that causes food to spiral as it moves through the small intestine, which really does help to increase absorption. We have some specializations of the muscularis externa. Of course, the smooth muscle in this layer are going to be important for peristalsis, for moving the food towards the large intestine, towards the anus ultimately. But there's another specialization here, and this is something called segmentation. With segmentation, we actually aren't just moving the food in one direction. We move it back and forth and back and forth. And it's very similar to a mixing type action, but what's called segmentation, because what we're doing is we're trying to break up or segment the food particles. So in this picture here, you can see how we have separate food particles shown in orange and blue. And through the process of segmentation, they get mixed together, and hence why they showed the end product as being green, because the blue and orange food particles were mixed together through the process of segmentation. Let's do a couple things with this slide. First of all, you're going to label the items indicated, but then there's also something that's a little bit confusing on this slide, maybe a misrepresentation, if you will, and see if you can figure out what that is. Once you finish the labeling and you've tried to find the confusing area, go ahead and hit play again. The thing that I found confusing is if we look towards the bottom of the slide, we see the terms submucosa and muscularis mucosa basically in the same area. And to me, that's confusing. You would think that, okay, this area must have two names, the submucosa and muscularis mucosae. However, the muscularis mucosae is referring to those yellow cells above it, right? So I just kind of found that area to be confusing. But other things we had to label were plica. Plica, actually, the labeling says if present, so it is not going to be present here because the plica has to be folds of the submucosa, and we barely see any submucosa here, so that's not present. The little mountain is our velus, so that's singular for villi. We can see microvilli on the absorptive cell over here. This layer of the mucosa is our simple columnar epithelium. The red and blue is our capillary, and then this yellow structure in between is the lacteal.